We all have a friend who gets along with everyone. Onion probably is that sweet friend among the vegetable. Got bored of the french fries? Go for the onion rings with your burger. How about some pickled onions to give that extra kick to your salad? Growing up, I could instantly tell by the smell of the onions that something delicious was being made in the kitchen, and that made my day. Georgia is famously known for this Vidalia onion industry, which contributes to a whopping 160 million to the state's economy. However, there is a growing concern in the onion industry due to a bacterial pathogen called Burkholderia. Despite being studied for many years, we still don't know how they make onion sick. Limited management options and a lack of clear understanding of the disease-causing mechanism means the bacteria poses a huge threat for economic loss to the onion industry. Through my research, we go back to the roots in understanding how these tiny microorganisms make onion sick. To do that, we look for the predictor gene regions in bacteria with the potential to make the onion sick and remove or mutate the component of those gene regions. We then check the modified bacteria to see if it still causes disease or kill the onion cells. There are at least 7,000 genes in the bacteria we study. So how do we know what genes to delete or mutate? We went fishing for the usual suspect first, meaning we selected genes that we know help many other closely related bacteria to kill their host. For example, one of our gene target was the bacterial secretion system or the type 3 secretion system. Think of this secretion system as a syringe or a needle. Bacteria utilizes this syringe and needle to inject the proteins that eventually kill the plant cells. We then deleted the genes that makes this syringe in our bacteria. Guess what happened? Surprise, surprise! Our bacteria could still kill the onion cells despite not having that syringe. Similarly, we removed many other suspects, but the bacteria was still able to kill the onion cells. So what is that X factor in bacteria without which the onions can't be killed? What looked like a straightforward question has now turned out to be a fascinating mystery for us. So the mechanism bacteria utilized to kill the onion cells could be more complicated than we initially thought. Through our research, we aim to understand the bacterial system better and unravel different strategies bacteria employ to win the arms race between them and the host. This is important in minimizing the crop loss and developing onion varieties that are immune to bacterial infection. That way, we can continue to enjoy onions while it still makes us cry.